Class.
Hmm? 5:30. షార్ప్ 4:30 స్టార్ట్ చేస్తాం. షార్ప్ 5:30.
Sure. I think. Last time.
Good evening to all. Yesterday we discussed about the carbohydrates. Yesterday we discussed about the carbohydrates. Today we are going to discussing about the electron transport chain. Besides the two pathways in the cellular respiration like glycolysis and citric acid cycle. Yesterday we discussed about both the glycolysis and citric acid cycle. The pathway which generates the ATP and the ATP generated during this aerobic catabolism of glucose is not generated directly from these pathways. So it is derived from a process that begins with moving the electrons. So during the moving of electrons, a series of electron transporters that undergo redox reactions. The electron transport chain, this causes the hydrogen ions to accumulate within the matrix space of mitochondria. Therefore, a concentration gradient of uh, forms in which hydrogen ions diffuses out of the matrix space by passing through the ATP synthetase, and the current hydrogen ions powers the catalytic action of ATP synthase, which phosphorylates the ADP and produces the ATP. Here it is a energy generation reaction. It is the last component of aerobic respiration and is the only part of glucose metabolism that utilizes the atmospheric oxygen, and oxygen continuously diffuses into the plants in animals. It enters the body through the respiratory system. Electron transport is a series of redox reactions that resembles a relay race or a barrel in between the electrons that are passed rapidly from one component to the other component to the end point of the chain where the electrons reduce molecular oxygen and produces the quantum. Therefore, four complexes composed of proteins and the aggregation of these four complexes together with associated with the mobile accessory electron carriers is called as electron transport. Here the electron transport chain is used to transport. It is presented in a multiple copies of the inner mitochondrial membrane of eukaryotes. In eukaryotes, the plasma membrane of, in case of prokaryotes, it is presented in the plasma membrane. And the electron transport chain in the prokaryotes that may not require any oxygen because it is happens in absence of oxygen in the anaerobes. As some anaerobes, this is the common feature of electron transport chain, which is which presence of proton pump, which creates a proton gradient across the membrane. So what happens in the complex one? Here you can see the complex one. When the two electrons of the complex chain are carried to the first complex bond abroad. Here you can see the NADH. This complex, which is labeled as 1 here, 1, which is composed of flavin mononucleotide FMN and an iron sulfur containing protein you can see here is this FMN, which is derived from the vitamin B2. And here the vitamin B2 is also called as riboflavin. It is one of the several prosthetic group of the, it is a cofactor which plays the major role in the electron transport chain. It is a prosthetic group which is having a non-protein molecule which are required for the the um, activity of the protein during the electron transport chain. And here, in case in the enzyme in complex 1, NADH dehydrogenase, name itself indicating that it acts as a dehydrogenase and is very large protein which containing the 45 amino acid chains. And a complex 1 can pump 4 hydrogen ions across the membrane from the matrix into the inner membrane space. And in the way the hydrogen ion gradient is established and maintained in between the two protein compartments. So here you can see the Q and complex 2 and complex 3 also you can see here. In both the complex 1, 2, 3, 4, in case of complex 2, complex 2 is directly receives the FADH2 and which do not pass through the complex 1. So here where this FADH2 that will not pass through the complex 1, the compound connecting the first and second complexes. Here this is the compound which connects through both the complex 1 and complex 2 to the third one, that is ubiquinone Q molecule, is lipid soluble and freely moves through the hydrophobic core of the membrane. And once it is reduced, it becomes like a QH2. Ubiquinone delivers its electron to the next complex in electron transport chain. Here the quinone complex, ubiquinone, which receives the electrons derived from the NADH from the of the complex 1 and electrons derived from the FADH2 from the complex 2, which includes the succinate dehydrogenase. This enzyme and FADH2 form a complex that delivers the electrons directly to the electron transport chain, which bypassing the first complex. And since these two electrons thus do not energize the proton pump in the first complex. And here the ATP molecules which made up of FADH2 electrons, the number of ATP molecules ultimately obtained after the reaction that is directly proportional to the number of protons pumped across the inner mitochondrial membrane. Here in case of complex 3, yes, the third complex 3, third complex is composed of cytochrome B and another ferric sulfur protein. Here the ferric sulfur protein center and the cytochrome C, this complex is also called as cytochrome oxidase reductase. These cytochrome proteins have a prosthetic group of heme proteins. The heme molecule is similar to the heme which is presented in the hemoglobin. This carries the electrons, not the oxygen. As a result of this, the iron ion and its core is reduced and oxidized as it passes the electron. That fluctuating in between the different oxidation states like Fe plus 2, which is reduced into Fe plus 3. The reduced form here is Fe plus 2 and the oxidized form here is Fe plus 3. The heme molecules in the cytochrome that have slightly different characteristics due to the effects of different protein binding them, which is giving very slight difference characteristics to the complex molecule. Here the complex 3 pumps protons through the membrane and passes through the electrons to cytochrome C from transport to the fourth complex of protein called cytochrome C, which is the acceptor of electrons from the ubiquinone. Here ubiquinone carries the pairs of electrons to the cytochrome C that can accept one only one time. So our next complex is 4. This is the complex which is composed of cytochrome proteins like cytochrome C, cytochrome A and cytochrome A3. This complex which containing again 2 heme groups. So here the one group of the 2 cytochromes, cytochrome A and cytochrome A3 and the 3 carbon ions called here is a pair of copper A and 1 copper B in the cytochrome A3. These cytochromes that holds the oxygen molecule very tightly in between the iron and copper ions, where until and unless the oxygen is completely reduced. Once the oxygen is completely reduced, then only the cytochrome hold the oxygen molecule very tightly in presence of oxygen. The reduced oxygen then pick up the 2 hydrogen ions from the surrounding medium to form the water molecule. The removal of the hydrogen ion from the system contributes to the ion gradient used in the process of chemoosmosis. What is chemoosmosis? The free energy from the series of redox potential reactions, just that is described in the now we discussed in the pumping of hydrogen ions across the membrane, the uneven distribution of hydrogen ions across the membrane, which establishes both the concentration and electrical gradients. Thus, an electrochemical gradient owing to the hydrogen ions, the positive charge and their aggregation on one side of the membrane. This membrane which open to the diffusion by hydrogen ions and the ions which tend to diffuse back across the matrix, which in presence of electrochemical gradients. So here the ions cannot diffuse through the non-polar regions since there is no hydrogen molecules or water molecule regions. So the phospholipid membranes without the aid of ion channels, which is very similar to the hydrogen ions in the matrix space that can only pass through the inner mitochondrial membrane through an integral part of the protein called ATP synthetase. This complex protein which can able to act as a generator, a minute generator which turned by the force of the hydrogen ions which that can able to diffuse through it. So it is also depending upon their electrochemical gradient. The molecular mechanism of this one is here the addition of the phosphate molecule taken place to the ADP. So the formation of ATP taken place. Using the potential energy here is hydrogen ion gradient. This is all about chemoosmosis. And we also discussed about the pump hydrogen ions. And what is oxidative phosphorylation? In your book, oxidative phosphorylation spelling is wrong. So please correct it. So here the oxidative phosphorylation means all the living organisms, yes, definitely, that can need oxygen uh, for their life. So without the oxygen, most of the living organism cannot able to respire. Uh, for holding breath for a longer time, it is very hard to living organisms. And lack of oxygen can makes the living organism feel very dizzy, and they feel like when you prolongly hold back inhalation, yes, definitely death may cause. So here, this is finding that every living organism definitely requires oxygen for their life. So cells, when need oxygen, that can use during the oxidative phosphorylation. The final stage of the cellular respiration, oxidative phosphorylation, which is made up of two closely connected components called electron transport chain and chemoosmosis. In the electron transport chain, just in the last step only, we discussed about the electron transport chain, how the electrons are transported in a different levels with the help of electron chemical gradient. Even in case of chemoosmosis also we discussed, the energy which is stored in the gradient is used to make the ATP. So here the oxygen is the end of electron transport chain where it accepts the electron and picks up the protons to form the water molecule. If the oxygen is not presented, the acceptor electrons, for example, because a person is not breathing in the enough oxygen, what happens? The electron transport chain will definitely stop running. Why? Because there is no, thank you, there is no oxygen, and ATP will no longer be produced by the chemoosmosis. Without enough ATP, cells cannot carry out the reactions. They need, they need to function, and after a longer enough period of time, definitely death may cause. Oxidative photophosphorylation are also known as OXPHOS, means O stands for oxidative, OX stands for oxidative, P stands for phosphorylation. That refers the redox reactions, which involves the flow of energy molecules, electrons, along a series of membrane bounded proteins, which is coupled with the generation of adenosine triphosphate. Oxidative phosphorylation is the fourth and final step in the cellular respiration, which the respiration can be represented as a simple reaction called combustion, where the combustion means the releasing of the energy molecule taken place, where the carbohydrates to form the carbon dioxide and water. Usually, when we fire the living materials like leaves, the combustion happens. There, the releasing of CO2 taken place because of the cells may get ruptured. There, the water molecule also releases. The same reaction here also happens, but there we are not using any fire. The cell which cannot afford to release all the chemical energy stored in the carbohydrates is a single step where the it is a reversible damage that intricate the balance and hemostatic of status of the organism. Here you can see the a simple reaction called if it is a C6H12O6 and CO2, uh, oxygen molecule, where the releasing of CO2 taken place along with the water and energy. So here the equation which represents the combustion of the carbohydrates. Here you can see the structure of mitochondria where the oxidative photophosphorylation taken place. ATP synthesis, the protein complex that harvesting the energy from the protein gradient and which using it to make the ATP from the 2-ADP. Like ADP is an enzyme that has the several names called in the complex 4 that protein translocating ATP synthetase and ATP synthetase central to the its function is movement of proteins taken place through it where you can see this reaction in the inner membrane spaces back to the matrix here in the matrix and if the ATP is available. Yes, in some cases for the concentration of the proteins that is presented inside the matrix than outside of it. When the reaction is happened, the ATP synthesis that run backwards with the proteins, there will be a moving from the inside to the outside that accompanied by the conversion of the ATP to ADP and phosphate also released. This is usually not a desirable way and there are some controls to reduce this reaction occurrence. Here you can see the electron transport across the inner mitochondrial membrane, which creates the proton gradient, which is used to fuel the ATP synthesis. So here, in the inner membrane, the electrochemical potential generated across this membrane. This is what happened. And in normal conditions, what happened? In normal condition, ATP concentration will be higher inside of the mitochondria and ADP concentration will be higher outside the mitochondria. So when the ATP synthesis which exceeds the rate of the ATP usage, then the ATP concentrations are raised outside the mitochondria and the ADP concentrations becomes very less. This is what happened. For example, during the the organism in the rest period, the overall effect of the reducing transport and lowering the concentration of ADP inside the matrix and reduces the ADP concentration inside the matrix. That reduces the oxidative phosphorylation and has affects the respiratory control. This is what happened in two cases. In the one case, it is not the favorable environment. This is not the desirable circumstances. Where in case of normal condition, what happened means ATP concentration will be higher in the mitochondria. In ADP concentration is very less in the outside the mitochondria. The next topic is, yes, here up to now we discussed about the glycolysis and electron transport chain and how the ADP and the ATP behaves in the oxidative phosphorylation and redox potential and ATP, ADP and ATP synthetase also we discussed. Next, our topic is how much ATP energy generated in the cellular respiration. Here in case of glycolysis, as we discussed, here you can see the structure of glycolysis, the pathway of the glycolysis in the cytoplasm. Here, the energy investment stage is the first level where the ATP energy involvement may taken place. In the energy harvesting stage, yes, the releasing of 2 ATP means here the net gain of the ATP is 2 ATP molecules. So here in the glycolysis, oxidation of one glucose molecule to two pyruvic acid molecules. There the production of 2 NADH plus H2 taken place, where the yield of the ATP in the substrate level phosphorylation, the 2 ATP molecules are released, and 4 to 6 ATP molecules are released in the oxidative phosphorylation in the electron transport chain. Whereas in case of mm, uh, the source here is a formation of two molecules of acetyl coenzyme A in acetyl coenzyme A in citric acid cycle. So there the formation of 6 ATPs by oxidative phosphorylation in electron transport chain. And in Krebs cycle and electron transport chain, the oxidation of succinyl-CoA to succinic acid happen where the 2 GTP that are converted to ATP. Here it is a substrate level phosphorylation, and the production of 6 NADH plus 6 H plus. Here the production of 18 ATPs. This is also a oxidative phosphorylation in the electron transport chain, where the production of 2 FADH2, there the releasing of 4. Here it is also a energy. Net gain here is 4 ATP molecules, where the oxidative phosphorylation in the electron transport chain. Overall, 36 or 38 ATP per glucose molecule. If one glucose molecule is involving, yes, approximately 38 ATP molecules are produced. This is the overall reaction. The energy change for the reaction can be obtained by the using of this formula called so yes, here the overall summary for this one, like carbohydrates and glycolysis, electron transport chain, is here all the living organisms, all containing the organic substances like carbohydrates, proteins, fats, and nucleic acids. Along with that, it is also having the minerals, presence of minerals like sodium, potassium, calcium, phosphorus, and iron. And carbohydrates are the main source of energy for living organisms. The substance like sugar, starch are some of the best examples of the carbohydrates. Generally, these are classified into 4 groups called monosaccharides, disaccharides, polysaccharides, oligosaccharides, and polysaccharides, which is depending upon the number of saccharin units presented in it. The term saccharin is which is coined for the some carbohydrates like sucrose, which is derived from the cane sugar, and glucose, fructose, which is because of their nature of sweetness, which is having the crystalline structure, and it is also soluble in water. Definitely, as everyone knowing, animals get the carbohydrates through their food. That is, even yesterday also I have given one example that these days people stopped eating rice because they are getting more amount of carbohydrates after eating the rice. So people are turned their views to use the millets. So as like that, here the carbohydrates are plays the major role for producing the sugar in the animals. So glycolysis, it is the metabolic pathway that converts the glucose into the pyruvate. Here the pyruvate or pyruvic acid and a hydrogen ion. Although 2 ATP molecules are used in the steps like in the glycolysis, 1-3 steps, and ATP molecules are generated in the 7th step, and second and last step, this gives rise to 4 ATP molecules. So overall, there will be a presence of 38, there will be a presence of 4 ATP molecules, which is generated after the glycolysis, where the broken down of or oxidation of pyruvate taken place for the releasing of carbohydrates in the cellular respiration, that is present in that is through the glycolysis. So here the primary source of acetyl-CoA is the sugar that is broken down by the glycolysis to produce the pyruvate. Besides these two pathways in cellular respiration, one is glycolysis, the other one is citric acid cycle that generate the ATP. This is about the carbohydrates. And our next topic is protein. Here, as everyone knowing that proteins is the staple food of the human body. Most of the people who is mostly depending upon the proteins. And here the protein that can be defined as the high molecular mass compound, which is uh, having a largely or entirely chain of amino acids. A group of amino acids uh, are called as proteins. Here, most of the cellular components or cells are composed of protein. All living organisms are mainly composed of organic substances, as just we discussed, like protein and amino acids and um, amino acids and fats. And here, besides this, yes, definitely, this depending upon the sodium, potassium, calcium, phosphorus, iron as a minerals. So this is the food, this is the requirement for the animals to get the energy for sustaining and growth and their body maintenance. Food is also mainly consisting of carbohydrates, proteins, fats, minerals, and vitamins. Here the proteins are the macromolecules. These are the polymers composed of amino acids. These are the basic unit of the life. It varies generally in molecular dimensions and structure and the presence of number of amino acids. Here, the proteins, there are 20 different amino acids presented throughout the world uh, for the construction of the proteins. Amino acids that are classified into two types. One is essential and the other one is non-essential. Name itself indicating that essential. The essential, those that cannot be synthesized by the animal body and has to depend upon the plants. So these forms the most important constituents of the animal body. They account for about 1/2 of the total dry weight of the body. Amino acids are the basic unit of the proteins, as everyone knowing. The basic unit of the protein is amino acids. So there are 20 different amino acids generally presented in proteins. Amino acids that are classified, it's essential and non-essential amino acids. Here the essential amino acid that cannot be synthesized by the animal body, has depending on the plants. And here we have the different amino acids like leucine, isoleucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, arginine, and histidine. Uh, Along with the methionine, uh, even for the hair growth also, we require methionine. And lysine also we require. And leucine also we required. Because of the presence of high amount of the leucine, someone gets the hair with wrinkles. So non-essential amino acids are alanine, and aspargine, aspartic acid, and cysteine, glutamine, and glutamic acid, glycine protein, and serine, and tyrosine. Here you can see the structure of the protein, which is presented like this. As we know, a group of amino acid, a link of amino acid forms the protein. So here in place of R, yes, so definitely we will get the substituent group or the functional group. Based upon this functional group, the type of the amino acid or the protein taken place. And here the COOH represents the acidic group, whereas the amino group represents the basic group. Depending upon the chemical nature, amino acids are classified like this. One is neutral amino acids with leucine, phenylalanine. This is not a phenylalanine, phenylalanine, cysteine, and acidic amino acids. Those were glutamic acid, aspartic acid. Name itself indicating like acid because of its acidic condition. And basic amino acids, lysine, arginine, histidine. Here the molecular number that may ranges from several thousands to several millions. Their polymer which is containing the carbon, hydrogen, nitrogen, and usually it is also containing the, if it is a cysteine, it contains the sulfur. Because of the high amount of the sulfur, as just I told you, uh, 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 okay, sulfur and phosphorus. Here the elementary composition of most of the proteins is markedly constant, about 53% of the carbon and 7% of the hydrogen and 23% of the oxygen, and also it contains 16 percentage of the nitrogen, 1% of the sulfur, and it contains very less amount, like lesser than the 1% of phosphorus. Here, what is peptide bond? Peptide bond means, yes, amino acids that is linked to each other to form a bond called peptide bond. Here, the peptide bond is a bond forms in between the carbon and amino group. Here, one CO and one NH that forms a bond. So amino acids that contain both amino group and carboxylic acid group in the molecules, and therefore which acts as a acids or alkali bases at the time, which is called as amphoteric. Due to these properties, amino acids unite each other to form a bond called dipeptide bonds and forming the large complex protein molecules. Here you can see the structure formation of peptide bond. Here the free amino group, uh, here you can see NH2, and here you can see the free carboxyl group. There the formation of peptide bond taken place in between the free carboxyl group and free amino group for after the formation of a peptide bond. And here, sorry, here you can see the primary structure of the insulin, a group of amino acids. Here you can see the formation of peptide bonds in between the amino group and carboxyl group. So when coming to the structure, as just we know discussed, the peptide bond is one of the combination of two amino groups. So it is formed by the combination of carboxyl and one amino group of one amino group which is presented in one amino acid, and the carboxyl group which is presented in the another amino acid. Together yield a water molecule, such bonds which contribute to form a protein polymer. Okay. Structure. When coming to the structure, here the primary structure, it is the sequence of amino acid which is arranged in protein molecule which is linked together by peptide bonds. It is the complete description of the covalent bond connection in the protein. So here in case of insulin, 51 amino acids you can see. Here in this structure, you can see the 51 amino acid. So this is the primary structure, which is a linear one, which is a linear structure. In case of secondary structure, where the folding of polypeptide chain into the coiled. The primary structure, in the primary structure, you can see the chain-like arrangement of the amino acids. Here, there is no uh, coils or the plate structure you will not see in the primary structure. Whereas in case of secondary structure, you can see the presence of foldings of peptide chains. That coil and plated structures held together by hydrogen bonds, or the foldings which is stabilized by the presence of disulfide bonds. So we can consider such uh, coiled regions are called as alpha helix and the beta helix, beta plated sheet. This type of arrangement you can see in case of skin fibrin and collagen, which is presented in the skin. Here I have missed a point. I just forgot it while I was telling. That is cysteine containing amino acid, that is neutral amino acid, cysteine, which contains high amount of the sulfur. Because of that reason only people, most of the people having curly hairs because of cysteine. Okay. And the other structure is tertiary structure. Here in case of tertiary structure, it is a long peptide chain with or without helix as like secondary structure, and it is presented like a many folded structures. You can see that because of many foldings, it is showing that three-dimensional view, three-dimensional configuration. It will show you there will be a presence of various weak molecular forces within the protein to for uh, big, for making the structure stabilized. Such forces here is van der Waals forces. And these are the forces which is presented in the disulfide bridges and ionic bonds. So the best example for this tertiary structure is globular protein. What happens in globular protein means, yes, every helix, it is a long, since it is a long chain peptide, every helix is coiled uh, in many times. When it is many times folded, it looks like a different dimensional configuration. So the quaternary structure, the union of different polypeptide chains, which is not linked covalently to give a functionally active molecular structure. Each polypeptide chain in such a protein is called as subunit. The subunits may be identical homogeneously. Quaternary structures are found. This type of quaternary structures you can see in the TMV virus, tobacco mosaic virus. And when coming to the classification of the proteins, yes, classification of the proteins mainly depending upon the molecular structure, chemical composition, and based upon their solubility and their function. Here the molecular structure, the fibrous protein which is presented mainly in collagen of tendons and myosin of muscles. and keratin of hair, and fibrin of coagulated blood. So these are the long and rod-shaped molecules of polypeptide chains, which are arranged or which are twisted together in a variety of ways to form the strands. This is the protein which are stable and capable of contraction and expansion. And the other one is this is depending upon the molecular structure. And globular protein, these are the proteins which is composed of globular and oval or spiral shaped molecules, which is soluble in water. And the example for this one is albumin of the egg, hemoglobin, enzymes, and snake venom, and the proteins which is presented in the scorpion, etc. So here the chemical composition of, depending upon the chemical composition of the protein, proteins are of three types. One is simple protein, conjugated protein, and derived protein. A simple protein, which is the protein which is of presented of the primarily formed of amino acids. Those were albumin, globulin, and globin. And conjugated proteins, these proteins, in addition to the amino acids, this is composed of, this is the combination of non-protein substances. Here the prosthetic group in their molecule. For example, the protein globin is combined with the iron containing porphyrin ring, which forms along with the heme to form the hemoglobin. Here the hemoglobin, heme is a protein, and here the combination of iron which containing the porphyrin compound, which forms the hemoglobin, which is the example for the conjugated proteins. And lipoproteins, which is presented in the egg yolk sac, and serum proteins of the serum proteins, and nucleoproteins, those were DNA proteins and RNA proteins, and mucoproteins, which is presented in the mucus of saliva, gonadotropins, and pituitary hormones, chromoproteins, which gives the color, and hemoglobin, hemocyanin, cytochrome, riboflavin are the examples for this one. And metalloproteins, the carbonic anhydrase enzyme, and tyrosinase enzyme. The derived protein, those were derived from the original proteins by the activity of the external factors like heat and the chemical agents. So here the derived proteins are proteases and peptones. Depending upon the solubility, some proteins are directly solubilized in water and some are not. So some are solubilized in the very mild acids, and some are solubilized in the strong acids and alkalis. So depending upon the solubility, here we have the water soluble proteins called albumin, and water insoluble and soluble in very dilute salt solutions like 95% of NaCl. Those were globulins, and globins, which highly solubilize in the ammonium hydroxide, and histone proteins, those were water soluble. And depending upon the function, we have the structural proteins and contractile proteins, enzymes, hormones, antibodies, and blood proteins. Here, structural proteins, the proteins which is efficiently participated in the formation of the different body parts, like more than half of the total protein of the mammalian body, which is composed of collagen, that is there in the skin, and presence of cartilage and the bone. And when coming to the other one, contractile protein, these are for contraction and relaxation of the muscle cells. Those are actin and myosin. These proteins are also presented in the unicellular organisms, not only in the multicellular organisms. Enzymes, these catalyzes the reaction. So this is also known as a biological catalyst. So this represents a large class of proteins. Nearly 2000 different types of enzymes are presented up to now, so called as biological catalysts. This plays the major role for any activity in the living organism. And hormones, when coming to the hormones, universally known hormone, that is insulin. Many of hormones are proteins in nature. Here, example like insulin, some other are steroids. And antibodies, these are the uh, antibodies which is generated from the human body when the foreign material like antigen comes into infectious material comes into the human body. So this is uh, the higher organism which produces the antibodies which destroys the foreign material. Here the gamma globulins are the best example. Gamma globulins are the examples of the antibodies in the mammals. Those were, as we know, uh, immunoglobulins. Here the immunoglobulins like immunoglobulin A, alpha, and we also having the M and secretory antibody and G, gamma. All those were the example for this functional antibody. When coming to the blood proteins, the albumins and globulins and fibrogens are the three major protein constituents of the blood. So what are the functions of the proteins? Here is, as just we discussed in the just last topic. Yes, some proteins definitely serves as a structural materials of the body. Those were myosin of the muscles, keratin of the skin, and keratin of the hair in the mammals, and collagen of the connective tissue in the skin. Most of the hormones produced by the endocrinal glands that were a protein in nature, most and some of peptides in nature, and some are derivatives of the amino acids. And enzymes are also protein in nature that catalyzes the many reactions in the living system. Hence, this is also called as biocatalyst. And proteins are essential from growth and repair of the animal tissue. These proteins are also provides the energy, heat that the body requires. The dietary proteins which provides the amino acids that required for the formation of the blood proteins, those were a serum albumin, a serum globulin, fibrinogen, and prothrombin, and conjugated proteins like globulins and hemoglobin. When cut happens in the when injury happens or cuts happens in the skin, because of this fibrinogen and thrombin and prothrombin, that converts the that stops the bleeding and forms like a mesh like manner. So this is the major activities of fibrinogen and prothrombin. Proteins in the form of immunoglobulins, just now we discussed. Yes, immunoglobulins plays the major role when the body defense mechanism is required. So of the body that acts as antibodies, which acts against the antigens. So here the proteins which functions in the defense mechanism of the body. And proteins forms nucleoproteins. Those were important constituents of the genes. When coming to this is about the introductory part of proteins and the protein classification and the structure of protein, how protein looks like. in what are the mechanism is performed by the protein, and uh, and what are the uses of protein, and where the protein presented, and what are the functions of the protein. When coming to the metabolism, the in animal, after the digestion of the food, the protein, the amino acids are absorbed into the blood bloodstream, where from the blood circulatory system it is reaches to the liver and to the other tissues. In liver, the amino acids can be utilized for the energy production in the process of amino acid broken down to the into the ammonia formed from the amino group, where one amino group which is reacted with the hydroxyl group forms into ammonia, where this amino group which is converted to the urea that will be excreted through the excretory system. And the other hand, the amino acids are also utilized for the protein synthesis and other biologically important substances. So here we have the topic called transamination. What happens in case of transamination? Name itself indicating that transamination. The first step in the catabolizing a broken down amino acid is the removal of the amino group for the formation of the ammonia. So amine group that can be transferred or removed through the transamination or deamination. Here the broken down of the amino acid and the amine group is removed. Deamination. D means removal, amination means amino group. So here the amino group are removed from the amino acid to a uh, keto group. So amino acid without the amino group, thus creating a new amino acid and keto acid. Here the keto group is C double bond O, keto acid. This reaction which is catalyzed by transamination enzyme, the family of enzymes called as transaminases. So the property of transaminases is removal of amine group. Actually, the transamination reaction that results in the exchange of amino group and one acid with a ketone group. Here the ketone group is C double bond O on the another acid. It is analog to the double replacement reaction. The most usual and major keto acid that involved with the transamino reaction here is alpha ketogluteric acid. It is the intermediate compound where yesterday we discussed about the citric acid cycle. So there also we have seen the ketogluteric acid. So that ketogluteric acid, alpha ketogluteric acid, which is a intermediate of citric acid cycle. So here we have a specific example for the transamination of alanine to make the pyruvic acid and glutamic acid. On the other hand, amino acids which can be converted after several steps through the transamination into the pyruvic acid, including serine, cysteine, and glycine. Here you can see the structure of alanine. Alanine, this one, CH3, COOH, and CH2, CH2, COOH, and COOH. And here you can see the alpha glutaric acid and pyruvic acid and glutamic acid. In this reaction, no ammonia or amino group is involved, and which is catalyzed by the specific transaminases which are believed to produce the muscle tissue. Such transamination really presented in the tissues. This enzyme that works only when glutamic acid or the aspartic acid or the ketone derivatives of these acids are presented in the exchange of amino group in metabolism. The amino derived from the deamination of the amino acid is first converted into glutamic acid, which in turn transfers its amino group to the keto, ketone acids by the process of transamination. Here, the enzyme which involves the important linkage in the joining of proteins, carbohydrates, and lipid metabolism. Here, the following reaction which shows that is alanine alpha ketogluteric acid forms the glutamic acid and pyruvic acid, and glutamic acid oxaloacetic acid which forms the aspartic acid and alpha ketogluteric acid. Here in the cells that can fix the ammonia by forming the glutamic acid from alpha ketoglutarate and glutamic acid. Here the glutamic acid is the fundamentally important in transamination reactions. So here you can see the amino acid carbon skeletons in citric acid cycle. They were the production of alpha ketogluteric acids by yielding the molecules that can enter into the glycolytic pathway, that is Krebs cycle. Here a single reaction that which is necessary for the conversion of alanine, glutamic acid and aspartic acid to form the uh, pyruvic acid and alpha ketogluteric acid and oxaloacetic acid. In addition to this reaction, there is a necessary formation of metabolic intermediates called other amino acids. Those were glutamic acid, glutamine, and histidine, proline, and arginine. Yes, this is a very important reaction, and it is also a reversible reaction. uh, since it is a reactable reaction, it links to the protein metabolism with the metabolism of carbohydrates and the lipids. Those amino acids that can form any of the metabolites of carbohydrate metabolism can be converted the glucose or the glycogen are called as glucogenic pathway. So here the amino acid that gives rise to the acetyl-CoA or acetoacetyl-CoA are said to be the ketogenic. Means here the ketone group is presented. Certain amino acid fall into both categories. The leucine, the only amino acid that is exclusively ketogenic. Here you can see the different types of the amino acid group. Okay, then tomorrow we'll discuss about the in the next class we will discuss about the next topic. I'm signing off.